Hello, my name is Alex Boyce, if you don't know already, and today I'll be presenting my Catalyst project. In this video, I'll be showing you what I did for my Catalyst project, why I made this Catalyst project, and how I accomplished my goals during this project. The reason why I picked a project this in-depth and this complicated was because I have always been fascinated by nature and the environment in which we live. For the longest time, I have been collecting and studying all sorts of creatures, such as fish, reptiles, and insects. As a kid growing up in a US Navy family, I was very fortunate enough to live in many countries around the world. In every location I lived within a short walk to the water, whether it was the Chesapeake Bay or the Mediterranean Sea. I was always looking to find interesting little creatures. In 2018, I conducted an Eagle project wherein I studied the migration patterns of a fish called the largemouth bass. Since I have conducted a study about fish before, I thought it would be very interesting if I conducted a study about the genetic variation of an invasive species of fish. So maybe, or possibly, I could tell if they spread by themselves or if they've spread by human introduction into new reservoirs around Singapore. As I conducted my preliminary research, I found that it requires a great amount of time and money in order to conduct a study about genetic variation. The reason why I chose a peacock bass as my fish to study for this project was because that they are an invasive species that is a popular sport fish to many Singaporean fishermen. Because these fish are of value to the Singapore fishermen, I believe that my MVP, my information that I gather at the end of my project, would value to them also, rather than just scientists. I discussed my problems with Mr. Kanufki, and we both came to a conclusion that it would be best to identify the strains of peacock bass in reservoirs around Singapore, because there had been recent reports of hybridization in the reservoirs all around Singapore. As I continued my research, I discovered a study in the 1950s about peacock bass in South America. It showed that there is less hybridization in warmer climates, typically towards the equator. And this is similar to Singapore, and a thought-provoking question occurred to me. Was hybridization really occurring in Singapore? Because of many reports and many visual identification, you could see that there was some sort of hybridization occurring. But I wanted to prove that with science. And that fueled my driving question. How could I conduct a study to determine whether these peacock bass are hybridizing or maybe even becoming their own subspecies? One of the most important things during the development of my project was making clear, successful connections and expanding my network. This was important because I could fully understand how to gain feedback and insight on what I could make better and what I could do better, so my project could run more smoothly and so that I had fewer errors along the road. In this section, I'll be showing you what a typical day of fishing to collect these samples is like. I'll be showing you how I collected a sample, and I'll be telling you my challenges that I had when collecting these fish. On the typical day of fishing, I would wake up at around 4:30 a.m., pack all my gear, and take a grab or bike to my spots. I would usually have one or two friends with me fishing early in the morning to different spots I went to, and I had a great time. Alright, so here we caught a little peacock bass. So this is something we're going to be taking a sample of. Alright, so what I'm going to be doing is I'm going to move this and I'll be putting it on here for the DNA sample. So I'll be taking out my knife and I'll just be slitting a small slit in its skin, just as if a bird nicked him. That's about all I need, so I'm gonna release him. And just so you guys know, none of the fish that I caught and took a sample of died during the process, so it was all fine. They swam off happy at the end. Even though it might look that I was successful every time I went fishing, I was not. Some days it came out empty-handed and did not have anything, and it's honestly, it's really just whether you get lucky or not and come across one. And how I solved this was that I kept fishing and I never stopped until I had all my samples. During the collection process of my project, I had a few successes. I caught many fish. I established a good relations with local fishermen. Yeah, and I caught my biggest peacock bass I've ever caught.
It was the best fight I've ever had with freshwater fish by far.
The next part of my project was to amplify the DNA to make sure that it amplifies so we could send it off to sequencing. So basically what I'm doing in this part is I'm isolating the DNA from the tissue, and I am basically amplifying the DNA from the tissue so that we can get copies and analyze the DNA. This process is very tedious, but at the end, the results were quite outstanding. In this part, I take the pure DNA and mix it with a little bit of primer and run it through the electrophoresis process.
Cool. So now it's going. All right. And so you won't be able to see much happening because it happens pretty slowly. But what you will see over time is stuff move through the gel.
As you can see here, we were successful enough to have 100% DNA amplification, which then we sent all of our DNA test tubes to get analyzed. Sadly, my results have not come in yet as of November 20th, 2020. It's okay though, because I will be updating my Catalyst document since I was given funding by the school. When I get my results, I will try to publish my research document so that maybe universities that are doing a project similar to this can work on it and really understand what I truly did, and maybe it could help them in a future project. For the last 2 weeks, I've been talking to my mentor's boss about having a QR code put up in a shop for locals to look at and to scan and to learn more about peacock bass in general. The QR code will be up in 2 places. It will be in this place called Fishing Paradise, where a lot of people go pay to fish in a pond where there's lots of fish. And there will be a code in a shop called Pro Kaiser Fishing Store. Over this project, I really learned that it takes a lot of time in order to analyze this DNA and collect all of these samples. You can't really predict when the fish are going to bite, and it's really interesting seeing how much effort it takes to just get a simple sample to be processed. Throughout this project, I realized that this is a future career path that I would like to follow throughout college, and I would love to learn how to analyze DNA and learn how to genetically engineer animals. I think that would be very, very interesting, very interesting thing to do. Although I had a few bumps along my Catalyst journey, I believe that I will truly remember this project as one that was very successful and one that I had fun actually doing. Thank you for watching my Catalyst video. Thank you for your time. Have a great day.